title of today's webinar is uncovering the functional potential of microbiome communities using shotgun metagenome sequencing analysis. Right. So yes, before I start this webinar, so I would like to let you guys know if you have any question. So feel free to type your question in the Q&A box. You can find them on the top of the team what this team apps. All right. So there's a Q&A button there, so you can type your question inside there, and please write your email so that if let's say we are unable to answer your question today, so hopefully we can answer your question by writing back to you by email. All right. Okay. All right. Let's start this webinar. All right. So just for presentation overview. Okay, so today we're talking about introduction about microbes and metagenomic, and then after that we'll be talking into the what is a shotgun metagenomic, some introduction, workflow, and what you can get from this metagenomic. Also some history about shotgun metagenomic, and after that we look into some of other application in the metagenomic, such as a shallow shotgun metagenomic, which is recently it's quite new, but we received a lot of interest from customer into this shallow shotgun metagenomic, as well as another one is such as a metatranscriptomic. Okay, we'll also look into that. And finally, we look into the metagenomic as a service by Apical Scientific and Berhad. Okay, all right. So as an introduction, microbes and metagenomic. So a microbe or microorganism is a microscopic organism that comprise either a single cell, a cluster cell, a cluster of cell, or multicellular, relatively multicellular, or it is a relatively complex organism. So it can found everywhere in soil, air, land, water, even inside the animal, inside even our guts, right? So microbes plays in a very important roles in everyday life. For example, let's say look into a fertile soil. So within this fertile soil contains billions of cells, microbes in the soil. So it is estimated that in a single gram of soil, there are several billions of bacteria alone. So, however, most of these bacteria are yet to be culturable. So it is reported that if you try to culture these bacteria, less than 1% of these microbes can be cultured. So, therefore, this culture approach is unable to accurately represent the entire of this microbiota in the environment. So this lead to a big vacuum in our standing on a complete microbe roles in the environment. Therefore, we need to have a different approach to study these unculturable microbes, right? So there are other than that. So microbes, the importance of microbes is we can use it to monitor the environmental health, such as in the agricultural. So we can study what are the microbes or bacteria that contribute to a better crop production. So, or in the food industry, for example, we can use it to monitor food processing to ensure high quality product. Or for example, in the research and development, so we're looking for a better enzyme, which have a better efficiency to be used in industry or in the food production. Right, so this is just a few examples of usage of microbe studies. There are many more. All right, so yeah, so let's look into what is metagenomic. So okay, the terms metagenomic metagenomic is an approach that is you to look at unculturable microbes. Started in 1980. However, during that time, sequencing is not that advanced. So this metagenomic approach is more on this functional based screening. So for example, like you can see in the figures, during that time, there is the scientists collect the sample from the environment, they isolate the metagenomic, and then they clone it to an expression vectors, and they express them to find their interest, enzyme interest, or any uh, microwave compound of their of interest. All right. So that time, the approach is kind of very slow, because you need to have a technical person, a very good trained technical person to handle this uh, lab. Uh, I mean, you need to construct the plasmid, you need to do the cloning, so you need to have a trained personnel to conduct this all this experiment. Not to mention the cost. You need to spend a lot of this to maintain the library. So cost, technical experience, trained personnel is important. So not everyone can have, not every lab have this access to this facilities. So yes, the metagenomic doesn't really work very fast that time. So yes. So just a little bit history about the shotgun metagenomic. Okay, so the shotgun metagenomic technically has started around year 2000. So by this guy, Craig Venter. So Craig Venter is considered as like somebody like Elon Musk in the molecular biology. So if you're interested about this guy, he's very interesting person. So if you can go Google this guy and read about his history. So he's a very willing to try out new approaches. Okay, so with the big funding that he has, so he started this what so called as a Sargasso Sea project, which is located near the Bermuda Sea. So what he did is he collect water from a few stations in this Bermuda area, Sargasso Sea, and he filter out the water and he collect the microbes. And using this, because during that time, Illumina or second gen platform is not yet available, because second generation sequencing platform is only available around 2007. So he sequenced all the samples using a Sanger Sanger platform. So he has to extract the DNA, clone it, and then transform in the cell, so that he can maintain all this metagenome library. So he had to sequence a lot of plasmid to get all these metagenome. So he had to spend a lot for this project. Okay, so this is the part of this finding, right? So so from this finding, from this project, he used a Sanger sequencing platform to sequence all these metagenome. So he sequenced about 1.66 million of sequence reads, which is equal to 265 MB. So we're talking about in year 2000, 265 MB is considered quite huge, because we sequence using a Sanger platform, right? It took years to complete this project. Right. So from these findings, he identified about 1.2 million previously unknown microbial genes. Right, 1.2 million is very huge. And from this 1.6 million of sequencing, he able to assemble them and managed to get a two complete genome sequence. Right. So this is a mixture of microbes, or we might call it like MAG. Right. So he managed to get a MAG of a Shewanella species as well as Burkholderia species from the marine sample. Right. Additionally, he also managed to find about 1,412 ribosomal genes, which is 148 previously unknown species. Apart from that, he also identified some eukaryote ribosomal genes. 18S ribosomal genes is very low coverage, right? Perhaps because he filtered the water is using a very small pore membrane, right? So that's why perhaps some of these large eukaryote have been filtered out. But nevertheless, he still managed to get some eukaryote from his sequence data. Also, interestingly, he also managed to get some viral sequence, which is about around 75 scaffold of fish genes. All right. Right. If you're interested to know about this project, you can go into and search for Sargasso Sea project John Craig Venter. So so there's a lot of things that been published inside the paper. You can go out and read it, find and read the paper. Okay. So now let's move on to the current era. So in year 2000, around 2006, 2007, the introduction of NGS revolutionized the metagenomic approach, right? So if as I mentioned earlier, prior with this NGS approach, you need to have a very technical person and also cost a very big budget to run this metagenomic analysis. You need to clone it, maintain the library, not to mention running the single sequencing, which is considered quite expensive, right? By having NGS in our to be accessible, right? So we can have a cheat sequencing, right? And then we don't have to clone. So because NGS is such as Illumina, you don't need to clone any of these DNA fragments. So you can just direct sequence from the extracted or any prepared samples, right? So this revolutionized the metagenomic approach. All right. So basically, we'll talk about this current era. So we can look into metagenomic approach. Basically, you can divide into two approach, which is we can have the Amplicon sequencing analysis and also metagenomic sequencing analysis. Right. So what's the Amplicon sequencing analysis? Basically, we'll just target a gene marker, for example, just a specific gene marker, for example, like cytin-S. So using a primer, so you amplify this gene marker from the metagenome, right, from the extracted metagenome, and you sequence and analyze them, analyze this data, right? So you will get a multi-copies of fragment from one target gene. For metagenomic sequencing, so from this extracted DNA, you need to shotgun the sequence, and after that you send for do a library construction and you sequence this sequence. So from this data generated from the sequencing, we'll try to reassemble back the data to reconstruct back the DNA metagenome, and finally we can try to do annotation and try to understand what is the contained in the DNA metagenome. Okay, so our first webinar series, we have covered the Amplicon sequence analysis for this, and for this second webinar, we'll be covering the metagenomic sequencing. So if you like to hear about my first webinar about Amplicon sequence analysis, you can go to our YouTube. So we have uploaded our first webinar series in our YouTube, and you can watch from there. All right. So basically, how this shotgun metagenomic works is very quite straightforward, right? So from the mixed microbial metagenomic, the first thing you need to do is you need to extract the DNA, and the DNA metagenome that you gain from here, you need to do a shotgun DNA. So the shotgun here means to fragmentize the DNA, because we for Illumina platform, so we unable to sequence the whole stretch of DNA, so we need to make all this DNA fragment to be smaller. So we have to shotgun, make it small pieces, and from this shotgun DNA, we'll do a library construction, and we sequence the library using the Illumina platform. Okay, and from this sequence rig that generated by this Illumina machine, we can process it and assemble. Right, so we can get the DNA assembled DNA metagenome, or we can try to reconstruct the MAG. So the MAG here it terms as a single taxon assembly based on one or more bin metagenome that has been a close representation of a certain microbe. So in other words, that we try to reconstruct back the microbe genome using the metagenome data, sequence data. Right. So from this data, there are downstream analysis that you can do. For example, taxonomy identification, functional information, statistic analysis, pathway analysis, comparison analysis, and many more. So for comparison analysis, is of course you can do it, but you need to have multiple samples. You need to have a set of different condition of samples, so that you can compare from sample A to sample B, and you can identify what are the genes or taxa that is been different compare one sample to another. All right. Okay, all right. So looking to this metagenome application, so there are a lot of application, as I mentioned in our earlier slide. Using a culturable approach, we only able to capture less than 1% of the microbes in the environment. So using a metagenome enable us to tap all these remaining 99% of the microbes in the environment. So, for example, in the research and development, in the search of a novel enzyme antibiotics, which is this is a very hot topic. For example, the antibiotics itself. Most of the antibiotic that is based on the cultured microbes. So, and we have a new issue on this, all this emerging of microbes, pathogen that is resistant to the antibiotics. So this is a race to find a new antibiotics, but we need to find them using a different approach. So perhaps the metagenomic enable us to find a new antibiotics. And bear in mind, all these antibiotics that has been in market is based on this 1% microbes that can be cultured. So just imagine how much potential that we can get from the remaining of 99%. So we might have an unlimited number of antibiotics that might be able to get from these unculturable microbes. So so this is some of this example of this shotgun metagenomic application, right? So for example, in the food industry, health sector, wildlife, university or local research institute, this is just some example. There are many more, right? All right. Okay, so for shotgun metagenomic, so what can you get from this data? So from the shotgun metagenomic, you can get like from this data, the sequence that you process them, you assemble, so you will get what they call a scaffold of the assembly. So from the scaffold of assembly, next, what you can do is you can do the annotation, and from the annotation you can do this taxonomy identification, which is, for example, as you show inside here. So you can identify what are the microbes that might be present, what are the microbes that might be dominant in a certain condition. Right, and with multiple of samples, you can do a comparative analysis. So, for example, like in the left C analysis, you can see what are the microbes that might be dominant in a certain in the respective sample, right? Apart from that, since we have this scaffold, we can do gene annotation, so we can look into what are the genes that might be present. So we can look into, like, for example, the CAC pathway, the core, we can identify what are the genes that might be present. So this might lead will lead us to identifying like new genes, new antibiotics, and so on. All right. Okay, all right. So that's about the shotgun metagenomic. Uh, yeah. So now let's move into this another type of shotgun metagenomic, another application for shotgun metagenomic, which we call it shallow shotgun metagenomics, right? Uh, Before that, for shotgun metagenomics, we need to have a kind of like quite a high coverage, and we need to have at least a minimum of 6 GB data to capture at least a good amount of the metagenome in the environment, right? So that leads to kind of like a kind of high cost there. So because you need to sequence quite deep coverage, right? So we have this another application where we call it a shallow shotgun metagenomics, which is similar to the shotgun metagenomic. I mean, in terms of the sample preparation, sequencing, they are similar, except that for the shallow shotgun metagenomic, you only sequence a shallow sequencing depth, which is about 1 to 3 GB of sequence data. Right, so you don't need to spend as much as you sequence on the shotgun metagenomic. So this lead to a reduced cost for sequencing, right? So what you can get from this shallow shotgun metagenomic is you can do it for study the taxonomy identification, right? So something is similar to Amplicon sequence analysis, right? So we can use it for taxonomy identification, but even better because if you use Amplicon sequence analysis approach, you only limit to a single taxa because always limit to the primer that you use for in the Amplicon sequence analysis. So if you use, for example, in the case of CTS, you want to target microbes, bacteria, for example, so you use CTS gene marker, so you only limit to that bacteria. I mean, like the text out. So you will you exclude like the virus, fungus, and so on, right? And also the primer also might will have some bias, especially well, I mean, because certain primer might prone to capture a certain group, but not to the other group, so they have some primer bias. Okay, so on the shallow shotgun metagenomic, you don't have you don't need to use a primers. So in other words, that you will capture everything inside this the metagenome, so you have enhanced taxonomy resolution, so you're not limit to a single taxa. The only limitation on this shallow shotgun metagenomic is the functional information will be limited, because unlike the shotgun metagenomic, you have a very high sequence depth, more than 6 GB. So this will help you to do a big good assembly. But for 1 to 3 GB sequence data, the assembly will be like fragmented, very short. Might not be enough to capture the functional information, but should be enough for you to capture the taxonomy, for you to do the taxonomy identification. So that's why for shallow shotgun metagenomic is suitable for you to look into the taxonomy identification, but not so much on the functional information. Right. So this is one of the publication where they compare the application quite similar. Okay, all right. Okay, so yeah, so just to do a simple comparison, so we do a slight comparison between the metagenomic and also shallow shotgun metagenomic, and also I include the Amplicon sequence analysis just to compare these three applications, right? To see how they are compared between these features, right? So for if you look at for the taxonomy information, you can look into this metagenomic and shallow shotgun metagenomic. They are very good because, as I mentioned in the previous slide, they don't limit to a single taxa, so you can cover like virus, eukaryote, or microbes. For Amplicon sequence analysis, the coverage is only limited to the primer that you use. All right. For the functional information, metagenomic able to catch a very good resolution of function information, but not for shallow shotgun metagenomic due to the low sequence coverage. And for the Amplicon sequence analysis, we will not be able to get any functional information due to this the gene marker. Right, the gene marker only carry the specific gene sequence. Right, not on the function information. So you won't get any function information if you use the Amplicon sequence analysis. Look into the sequence coverage for metagenomic. You need to have a high sequence coverage, as you need to have a minimum of 6 GB. All right. For shallow shotgun, you need to have a minimal sequence coverage, which is about 1 to 3 GB of sequence data, sequence coverage. While for the Amplicon sequence analysis, you only have a very low coverage. You even less than 1 GB to do the Amplicon sequence analysis. This lead to the cost that need to run this what do you call this analysis. For metagenomic, you need to have a high cost. Shallow shotgun, not so much, 1 to 3 GB, and for Amplicon sequence analysis, which is quite low, right? And for PCR bias, there's no PCR bias for metagenome and shallow shotgun metagenomic because there's no primer involved for this application. For Amplicon sequence analysis, yes, there is some PCR bias due to the type of you want to target what taxa, and also the primer might prone to cover specific microbe groups, not the others, right? So there's some PCR bias, right? And look into the host contamination. Metagenomic and shallow shotgun, there are possible host contamination, especially working with the host-derived metagenome. For example, you work with the animal guts. If you are extracted too much of the host sample, then for the sequencing, that you might have a host contamination in your sample. Right. So this is something that has to be careful, especially working with the animal guts. Processing the sample is very crucial, so not to have contamination. To limit, it's impossible to have remove this host contamination, but we try to limit the host contamination as much as possible. For the Amplicon sequence analysis, you can remove the host contamination by using this PCR. So the PCR can target the microbe, and you can exclude the host contamination from the sample, right? So it depends on your objectives and also your budget, of course. So if you want to select which approach which is that is suitable for your study, so whether it's metagenomic, shallow shotgun metagenomic, and also Amplicon sequence analysis. All right. Okay, so want to move on into another type of application, which is metatranscriptomic, right? So metatranscriptomic is another type of metagenomic approach, but rather than looking at the DNA metagenome, metatranscriptomic, they're looking at the RNA from this metagenomic sample, right? The approach is quite similar to this to the metagenome. So you can see from here, you have from the environment sample, you collect the harvest, you isolate the sample, isolate the microbes, and then after that you do the for DNA metagenome, you extract the DNA and you sequence them. But for the RNA metatranscriptome, instead of you extract the DNA, for metatranscriptome, you extract the RNA, proceed with the RNA depletion, and you sequence the mRNA. Then you get the mRNA raw reads. So from the mRNA raw reads, you can analyze them. So from the data generated, you will get the metatranscriptome assembly. Okay. Right. So metatranscriptome, in other words, is a profiling of community-wide gene expression. It's similar to RNA-seq, but instead of you looking into a specific experiment, like specific microbes culture, or perhaps in the animal work, but for metatranscriptome, your source of samples came from the metagenome sample, right? Metatranscriptome used to study the function and activity of a complete set of transcript from the environmental sample. So using this approach, you can look into the gene activity diversity. You can also do a diffusion express gene using a different set of microbiome communities. All right. For example, let's say you want to study the fermentation, for example. So you have a different type of, let's say, a formula. You want to see which microbe that tends to behave, tends to react well to this type of what you call this formula. So you can see what how they perform, and from there you can harvest the microbes, you extract the RNA to see what are the genes that might be expressed that might lead to this what they call this performance of the microbe in the formula, right? And this also you from the data generated, you can look into the functional and pathway diversities, right? One thing that's what is more why metatranscriptome is a very kind of like advantage here is because using this approach, you can use to start to study what are the most important functional pathway needed in the environment, right? So just for example here, so this is the one of the paper, right? There's many papers that have been published, which is using a metatranscriptome approach. So I just select one of this, which is they try to find look into this metatranscriptome approach to find genes that can use to degrade plastic. So the source of microbe is from the marine microbiome, and from this metatranscriptome approach, they managed to find a few genes that might potential to be used as industrial enzyme to degrade the plastic. Right. So this is one of the example that you can get from this metatranscriptome analysis. Okay. So if let's say we use the culture method, this approach will not be, this study will not be available. I mean, like we are unable to access all these findings. So that's how strong this metatranscriptome technique can be used. All right. Okay. So so that's about metatranscriptome. Okay, so now let's move into this metagenomic service by our company, Apical Scientific Sdn Bhd. So Apical Scientific Sdn Bhd, we can cover all type of metagenomic analysis, whether shotgun metagenomic, shallow shotgun metagenomic, or even metatranscriptomic. All right. So of course, if you want to send the sample, so it's very important. You need to have a very good quality DNA or RNA. So concentration, high concentration is a must, especially if you're working with shotgun metagenomic as well as the metatranscriptome. The concentration is very important, right? For sequencing coverage, you need to have at least about 6 GB data for the metagenomic and also metatranscriptomic. And for the shallow shotgun metagenomic, we do it for about 1 to 3 GB of shallow shotgun metagenomic. Okay, so about the sample requirement. So for this is a sample requirement required by Apical Scientific, right? If you want to go for the metagenomic analysis, so this is also applicable for the shallow shotgun metagenomic. So for the amount of DNA metagenome, so about you need to have about 200 nanogram, and the volume must be more than 20 microliter, with concentration of more than 10 nanogram per microliter. So the purity must be around 0.2. Sorry, OD of 260 over 280 must be about around 1.8 to 2.0. So we expect we won't see any degradation and contamination in the sample. For the metatranscriptome, so we need to have a total RNA. So on the amount, we need to have about 2 microgram of sample, right? The reason why we need to have a very huge amount of RNA samples because we need to do the ribosomal depletion. So that's really required a high amount of RNA sample. Okay, and for the volume, we need to have about 20 microliter and above, with concentration of 50 nanogram per microliter. The RIN integrity number, which is very important if you're sending any RNA sample. So RIN RNA integrity number must be very good. We expect to get like more than 6.5 with a flat baseline, right? And for the purity, similar as the DNA. So we need to have an OD of 260, 280 of more than 2, and 260 and 230 also above than 2, right? If you have any difficulties of getting a good quality sample, so we also have a DNA extraction service. So what you just need to do, just send the sample material to us. We can do DNA extraction for you. So we have experience doing genomic extraction, RNA extraction. For genomic extraction, for example, we may have a lot of experience extracting genomic material from soil sample, even from other source of metagenome. So so we have a lot of experience dealing with extracting DNA from metagenome sample. All right. So lastly, we'd like to bring you to our demo report for shotgun metagenomics. So we'll go briefly on this demo report. Right. So this is our demo report, metagenomic demo report. So if you can see over here, this is our Nova Lifetime, which is our bioinformatic partner, and which is our partner on analyzing all the bioinformatic data. All right. So we can have a look on the first page. All the data, all the results are being kept in all these tabs over here, which is you can click. All right. Right, so on the first page is just overview about metagenomics. So what's metagenomics all about, and also we'll talk about describe about the analysis workflow, how the sample will analyze. Right, so all the list of software used to analyze the data are being listed here. And then the next we have a figure of overview of metagenomic analysis workflow. Okay, and then we have the list of software for statistics analysis use, and as well as with the reference. Okay, all right. And next we have the statistic. So this is statistic for the like the assembly for the count data. So this is for your information. This is demo report based on 6 samples. So there are 2 groups. So each group have 3 replicates. We have A1, A2, A3, and then the second group will be B1, B2, B3. So this data has been analyzed, and then we also have a combine from all the samples, so which is represent inside this. So we have the assembled data comprising from all 6 samples over here. So we have the ORF prediction, CAG, ribosomal, all the details that been found from this assembled data are being listed here. Okay, and the next up we have a taxonomy. So we can see the taxonomy information. So what are taxonomy that identified from respective samples? All right, so our demo report over here, they are not, all the figures here are not the passive figure. Actually, they are interactive. So if let's say if you move your mouse over the bar chart, it help you to explain or extract the information that contain in each sectors. Right. So this will help our customer to view the information, especially when the bar chart are very complex like this. All right. Another features from this bar chart is that our legend here is also as there's some features on the bar chart over here. So for example, if try to find one example. So for example, if you're interested to know the abundance of this the taxa in this on the sample, for example, you're interested to know the virus present in the sample. So what you can do is just double click this. They will uncheck the rest of the microbes and only highlight the one that you select. So from here we can see the presence of this virus in across all samples. So they will help our customer to view the result easily. Right. So if you go back, if you double click again, so we'll turn back to the original state. Okay. All right. So let's try one more. Yeah. So we can select right these features. And you can find the you can highlight the abundance of the microbe, so you can compare across all samples. Right. Apart from bar chart, we also provide the result in the Corona chart. So Corona chart is very useful. So also it's very interactive. So you can play around. So you can, like for example, if you're interested to know the abundance of eukaryota, so you can just click here and then you expand it. So this Corona chart only focus on the eukaryota, and if you click on the legend over here, so each of the Corona chart are represented by all all the samples have their own Corona chart. So if you click this, so they will move it to the different sample, right? So like as I clicked earlier, so if you select the eukaryota, so the remaining sample also will present the result in eukaryota level. Okay. Apart from that, we have like result for KAGG, so we can show what are the enzyme that may be present, and the data, the number here represent the amount, the abundance of this enzyme present across all samples. All right, so we have the KAGG result based on the TPM and also RPKM. So based on the abundance of this enzyme present across all sample. Apart from KAGG, we have a COG, right? So same as the KAGG, show the engine annotation that present across all samples. Right. Apart from that, we have the alpha diversity and also the beta diversity. So we have for alpha diversity, we have few indices, right? For each, which all the indices have their own, what I call features, to explain about the samples, the metagenome sample. So same goes for the ordination. We also provide ordination. So we have a CCA, MDS, NDS, PCOA, and also RDA. So this is the beta diversity or ordination. Right, so we have also reflection. So from this reflection, we have based on the KAGG, COG, and also TEXA. So they explain how well these are being captured in the metagenome. Are we covering enough? So from this data, we can say that we can see all the graphia-rich plateau. So means that we covered most of this KAGG, COG, and so TAXA in this metagenome sample. Right, so there are a few more like the correlation analysis, heat map, Venn diagram, and LEFC. So which is LEFC also is one of the one of the analysis used to compare the microbes across. Want to see what are the microbes that are being dominant compared to another. So we run using a LEFC analysis. Right, so we can, for example, let's say we look at the taxa LEFC. So based on this let's see, we can see we can identify what are the microbes that has been uniquely enriched in respective groups. Okay. All right, so that's the last about the last page is about our company, Food Space, and so Exil. And this is the last one is our partner, BI Bioinformatics partner, Monova Life Tech. All right. Okay, that's about our demo report. Okay, so my presentation might not covered everything. So if you have a further question, so you any inquiry, you can send email to ngs@apicalscientific.com. So we'll do our best to answer your question. Right.